Forensic serology, the nature of blood. So blood is the fluid that circulates throughout the body by way of the heart, arteries, veins, and capillaries. And it transports oxygen, electrolytes, nourishment, hormones, vitamins, and antibodies to tissues and transports waste products from tissues to the excretory system. So in this lecture, we will discuss the composition of blood, which include plasma, erythrocytes, leukocytes, platelets, and then we'll also discuss the presumptive blood test lab. So blood consists of a fluid portion, which is called plasma, and then suspended in this plasma are cellular components consisting of red blood cells, white blood cells, and platelets. So the amount of blood we have on average, blood is 8% of our total body weight, and males have about 5 to 6 liters, and females have about 4 to 5 liters. If you lose 40% of your blood volume, this would produce irreversible shock and lead to death. The plasma is composed principally of water. About 90% is water with a pH of 7.4. And plasma accounts for 55% of the blood content. The main constituents other than the cellular elements are blood electrolytes, which are ions of inorganic salts dissolved in the blood plasma, and this maintains the osmotic balance in the blood, which explains the importance of Gatorade and other drinks such as that. There's also various plasma proteins, including lipid binding proteins. There are antibodies, also known as immunoglobulins, fibrinogens, which are clotting factors, various plasma proteins, which buffer against pH changes, so we want to maintain our pH of 7.4. And they also maintain osmotic balance, and as well as the blood's viscosity. And plasma transports other materials such as nutrients, metabolic waste, respiratory gases, and hormones. And the cellular components comprise the other 45% of our blood volume. And the main cellular elements suspended in plasma are red blood cells, also known as erythrocytes, white blood cells, also known as leukocytes, and platelets. So erythrocytes, also known as red blood cells, they transport oxygen from lungs via the arterial system, and they return carbon dioxide to lungs for expiration via the venous system. There's about 25 trillion erythrocytes in 5 liters of blood. And interesting to note, mammalian erythrocytes are anucleated, or they lack a nucleus. And they believe this evolutionarily why this occurs is so it provides more space for hemoglobin, and hemoglobin is an iron-containing oxygen transport metalloprotein. So the hemoglobin is what is in red blood cells that actually transport oxygen. And within one red blood cells, there's actually 250 million molecules of hemoglobin, and each hemoglobin binds to 4 molecules of oxygen. So a single blood cell can transport 1 billion molecules of oxygen, and you think about the number of red blood cells we have, that's quite a bit of oxygen that's transported. Other things to note, there's 4 subunits, each with the heme group, and that's what binds the oxygen. And it's a tetrameric cooperative binding, so when one binds, it makes the other ones easier to bind. And it's, I mentioned 4 subunits. And it also binds carbon monoxide. Well, what's different, or what's dangerous about carbon monoxide, it actually binds to the same site as the oxygen binding site. And what's interesting is that hemoglobin has a preferential binding for carbon monoxide as compared to oxygen. And it also binds carbon dioxide, but it binds at a different site. Leukocytes, which are white blood cells, and there are 5 major types of leukocytes. are monocytes, neutrophils, basophils, eosinophils, and lymphocytes. And these assist with defense against foreign substances and infection, and these are nucleated. And we're going to talk about the importance of why they're nucleated. And most of our leukocytes reside outside the circulatory system, and they're in the interstitial fluid and the lymphatic system. So there are about 5,000 to 10,000 leukocytes in a microliter of blood, but during an infection, this number temporarily increases. Platelets, or also known as thrombocytes, are blood clotting factors. So what they do is they convert fibrinogen to fibrin, and these are the major components of clotting mechanisms of blood. And then so the blood clots when the fibrin traps and meshes the erythrocytes. And if the clotted material was removed, you would have a yellowish liquid, and this is known as serum. And we're going to go more into what serum is. And what happens in hemophilia lowers the blood plasma clotting factor levels of the coagulation factors needed for a normal clotting process. So what happens when a blood vessel is injured, the temporary scab does form, but since the coagulation factors doesn't allow fibrin to form, you can't maintain that blood clot. The hemopoiesis or hematopoiesis is the formation of blood cellular components. So all blood cells, cellular blood components, are derived from the hematopoietic stem cell, which is a multipotential cell, or hematocytoblast. As you can see, this is the lineage, and this is where you get all your white blood cells here, right here, and then there's the red blood cells, and then here's your red blood cell right there. So the presumptive blood test is a presumptive test for the presence of blood. It's a quick and dirty test to see if there's actually blood. So what you are looking for is either pink Q-tips, or you use aluminol spray to see the glow. And one of the difficulties, it doesn't distinguish between human blood, animal blood, and horseradish. And we'll go into that. And it's used to find blood, and then you collect it for later analysis. So the test we're going to do is called the Kastle-Meyer test. And what it does is changes from an alcohol to a ketone in the presence of an oxidizing agent, and which is the oxygen from hemoglobin. So the Kastle-Meyer test detects the presence of enzymes in blood, and other substances such as potatoes and horseradish actually contain that enzyme peroxidase, which reacts with the phenolphthalein. So what happens is the positive Kastle-Meyer test is not definitive, and it only indicates the possibility that there's blood. And then you'll need to use other tests, and we'll go into that, which is the precipitin test, to verify the presumptive results. So what occurs in the phenolphthalein test is the big P is the active form of phenolphthalein, and the little p is the inactive form, and it's actually known as phenolphthalein. And what happens is when you have hydrogen, you add hydrogen peroxide, so you have phenolphthalein, and then you have hydrogen peroxide. What the heme does, it has peroxidase activity, so it breaks down that hydrogen peroxide that you're going to add into water and oxygen. And this oxygen goes into oxidizing our phenolphthalein, which is also known as our Kastle-Meyer reagent. It gives it this bright pink color. So you can see phenolphthalein plus a base, and when you add oxygen, when it's oxidized, then you get an P active, which is the phenolphthalein, and that actually comes out pink. Luminol, it's also a presumptive blood test. It's the one where things glow. It works in a similar fashion to phenolphthalein. except rather than getting a pink color, you detect with chemiluminescence. And the chief catalyst is iron, and it's not the catalase. And since you could use it to test a large area at once, as opposed to the thaline or the Kaplan-Meier test, and it doesn't degrade DNA or the blood antigens. And just like the Kaplan-Meier test, it fluoresces in the presence of horseradish as well as certain bleaches. So if a crime scene is thoroughly cleaned with a weak bleach solution, so the bleach will cause the entire crime scene to fluoresce. And it's an example of a room, and here's being tested with luminol. So the ABO classification was discovered by Karl Landsteiner, who also discovered the RH factor, and we'll go into that. So there are over 100 blood factors, and no two have the same blood factors except for identical twins. However, and then what blood factors are these antigens, which are these glycoproteins on the surface of red blood cells. There's over 100 different glycoproteins that are on the surface of the red blood cells, and this is what is responsible for the different blood types. However, the ones we focus are on these two antigens, the A and B. So the ABO system focuses on the two types of antigens, and then so if you have type A blood, you have A antigens. If you have type B blood, you have B antigens. If you have type O blood, you have neither antigens. And if you have type AB blood, you have both A and B. And 80% of the population are known as secretors, and what they do is they actually secrete the blood type antigen in other body fluids. So as you can see, in type A, there's A antigens present. Type B, there's B antigens present. Type AB, there's A and B antigens present. And type O, there's no antigens. We're going to go into antibodies later, but as you can see, in A, there's anti-B, in B, there's anti-A. Since you have both A and B, there is no anti-A or anti-B. And in group O, since there is no antigens, there's anti-A and anti-B. We'll go into that in a second. In addition, there's another factor called the D antigen. So it was designated as the Rh factor to indicate that the rhesus blood has been used for the production of the serum. So what we have are Rh positive, people with an Rh factor, and then Rh negative, people without the Rh factor. And it's approximately 85% are Rh positive. So this is where you get the A plus, and then the negative would be the A minus. So that's where you get the plus and minus. So as we were talking about serum, this is the blood plasma without the fibrinogen, but it contains antibodies. So what antibodies do are these proteins that are produced to attack invaders in the bloodstream. So if you have type B blood, you shouldn't have, if you have type B blood, then A blood would be an invader. So you would need to make antibodies against A. So that's why it's known as anti-A antibodies. And if you have type A, you shouldn't have B blood, so you make anti-B antibodies. If you have type AB blood, you have both A and B, so you wouldn't make any antibodies. And if you have type O blood, since you don't have either A or B, then you would make both A and B antibodies. And what happens is, this is an antibody, and they're bivalent, so they have these two valent sites, one right here and one right here. These are the antigen binding sites. And what occurs is when you get the wrong blood type during a transfusion, since they're bivalent, these two arms can connect. And since they could connect with, in this case, these are anti-B antibodies, they won't connect with A. But here they will connect with B, and since they have two arms, they could connect in any manner that they choose. And what you get is this clumping together also known as agglutination, and this is what what we look for in all of in several of our labs we do regarding blood. And so this is important in blood donation, where we want to match it correctly. And so what we have is type O blood could give to type A, type AB, and type B. Type A blood could give to AB, and type B blood could give to type AB. But AB could only give to AB. A could also give to A, B could give to B, and O could give to O. So O is known as the universal donor, since it could give to all three, and AB is known as the universal recipient, since it could receive blood from all three other types of blood type. As we were saying, blood type is determined by the proteins that are found on the surface of red blood cells, and proteins are made by genes. So we have 2 copies of each gene that determines the proteins. In many cases, as you learned in biology, genes are usually dominant or recessive. And so there's 3 forms of genes for blood type. There's no make A protein or B protein or neither protein gene. So in this case, we have capital I is gene, it's the gene, in the superscript form for so there's capital A, which is the A protein, capital I, capital B, which is the B protein, and lowercase I, which is no protein. So these are the genes. You can have the A protein, B protein, or the no protein. We'll go into the capital and lowercase in the next slide. So the A and B alleles, so there's 2 copies of each gene, and the combo determines what kind of blood type. So it determines what kind of protein you're going to make, and therefore your blood type. And why there's capital IA and capital IB is because A and B alleles are co-dominant. So one is not dominant over the other. So both are dominant over the lowercase I, which is the one that makes no protein. So as you can see, blood type A could have capital IA, capital IA, because these are both, they're homozygous, or you can be heterozygous, but you need the recessive, which is lowercase I. And then in the case of B, it's capital IB, or capital IB, lowercase I. For blood type A, you need to have both alleles, one for A and one for B, so capital A and capital IB. And to have blood type O, you need to be homozygous recessive, so you need to have lowercase I and lowercase I.